Welcome to the set of videos on plant-based survey planning implemented by ANR. In the EU-funded project, we have developed some tools for selection of quarantine pests for monitoring. The tutorials provide a quick overview on how to do this. In 12 videos, we explain the legal provisions behind. We give guidance for using different tools and sources of information to early detect the new pests. Those are non-native plant pests on growing plants, either in production or in nature. Such surveillance should be organized by official plant health services in the EU member states and also in EU candidate countries. The aim is to demonstrate plant pest freedom to trading partners and to be able to enter the EU market with plants and plant products. In this tutorial, we will explain how to initiate annual survey planning. First, we need to identify priorities for phytosanitary actions. We need to select among numerous plant pests and focus to the category of quarantine pests, which we would like to detect in official surveys. Quarantine pests are those non-native pests which are listed by legislation as quarantine. They may not be present, or if they are present, they occur in limited area and are under the official control. Such as this Asian longhorn beetle on the photo. Anoplophora gabrypenis is a priority pest for actions. If it is found, it has to be eradicated as it can destroy deciduous forests, parks, and fruit trees. It has an unacceptable impact to plant health, economy, and society. During the visual checks of host plants, we could observe many common pests which might be controlled by growers, but this category is not a subject of official surveys. Official services want to protect plants and other life on the planet by its action, which requires a scientific approach and a state budget behind. The pests such as Anoplophora gabrypenis are categorized as union quarantine pests. We would like to protect plants against them. Its cousin, Murimus funereus, from the same family Cerambicidae, would also go extinct in case of quarantine pest spread, as it is already now endangered and on red list of EU Habitat Directive as a protected species. In planning the risk-based surveys of regulated pests, a national plant protection organization has to define the scope of official surveys so that the necessary work could be properly justified and explained to hierarchy. In the set of tutorials, we will focus to planning the risk-based surveys for the quarantine pests, including what tools may help you in selection of the pests for the surveys and how to do it. In selection of pests for official surveys, we rely on the EU Plant Health Law, which is Regulation 2031 on protective measures against pests of plants, and its implementing regulations. Official services need to focus on the most harmful pests, which are defined as union quarantine pests. And how many are those? In the global database of EPA are more than 55,000 of plant pests. Out of all known pests around the world, 1,000 are regulated in the European Union as quarantine pests, and those need to be actively under the surveillance. Based on the recommendation of EFSA, 20 most harmful quarantine pests are listed as priority pests. The lists are published in both regulations. And annually we need to select also other pests to be surveyed. And why is that? Prevention and early detection of the presence of pests is extremely important for timely and effective eradication. To actively monitor plants for presence of thousands of quarantine species, we will need to have a crop-based approach and prepare a multi-annual survey program. The EU Plant Health Law allows to make up to 10 years programs in which the pest species rotate based on risk. Quarantine pests need to be surveyed by the competent authority, except those justified for exclusion. National Plant Protection Organization defines a scope of each survey as regards the area concerned and the time scale covered, as well as the pests which are on the plants and commodities targeted. Detection survey is done by visual examination, sampling and testing, covering the host plants in the country in the appropriate time to discover it. If the EU member states has also a protected zone for quarantine pests, this needs to be surveyed annually. This is required also for priority pests, except those justified for exclusion. In decision-making, which pests will become a part of annual survey program, we need to consult published regulations with listed pests. There are many. Some of them are not yet listed as quarantine pests, but they are regulated as they preliminarily qualify for quarantine measures. Here you consult Regulation 1090/941. The main quarantine lists are in Regulation 2072. In its Annex 2, Part A, we have quarantine pests not known to occur in the European Union, and those are of categories bacteria, fungi, and oromycete, insects and mites, nematodes, parasitic plants, viruses, viroids, and phytoplasmas. And the same is in Annex Part B, but those have somehow priority for the surveillance because they are already known to occur in the Union territory, and they have been found on limited areas, and emergency measures were adopted against them. The cases of such quarantine pests are Xylella fastidiosa, Anoplophora chinensis, Anoplophora glabripenis, Aromia bungi, Popillia japonica, and many others listed as quarantine pests, or emergency measures are established based on Article 31 of Plant Health Regulation. A long list of quarantine pests for detection surveys can be made shorter by justified exclusion of regulated pests. The surveys are not required for pests for which it is unequivocally concluded that they cannot become established or spread in the member state concerned due to its ecoclimatic conditions or due to the absence of host species. The surveys are though not required for pests which can be excluded from based on the justification, like host plants are not present in the natural environment, or host crops is not grown in the territory, or ecoclimatic conditions do not allow its establishment. This is justified in accordance with Plant Health Regulation Articles 22 and 24. For each of selected quarantine pests, a survey protocol has to be designed based on scientific knowledge of biology and epidemiology and available technical data on host plants. The detection surveys should take place in all areas where the pest concerned was not known to be present. So for the example of quarantine pests on pepper, we need to know the size of pepper population, that is, how many plants are grown in the country, for example, all paprika grown in 1 year. Then we select risky habitats where it is most probable that quarantine pests would be occur. We define epidemiological units. In this, we further select survey sites and define the inspection units where targeted visual examination and sampling will take place. This phase is prepared at the National Plant Protection Organization's headquarters, with assistance of scientific and technical support at institutes or universities where the risk assessors work. For the field surveyors, we need to prepare practical instructions from the protocol that they will be able to organize field surveys in appropriate time and place, and know the scope of visual examination and the number of samples that should be taken. For each selected quarantine pest, we prepare such a description as it is shown here for Alstonia solanacearum. We describe the type of survey, detection survey, describe references if we have other information on this survey protocol, describe the common name, potato brown rot disease, and host plants which are in the survey. Then briefly show the symptoms of infection and describe in the epidemiological units where actually the visual examination can take place. For example, for this disease, the horticultural reproduction of potatoes will take place. This is known in non-agricultural plants. It will be also checked in the warehouses. Together we can have a certain number of samples. And what is also important, there are inspection units and timing when this will sample. For example, a sample unit of 200 tubers for one sample. Then complete plants of tomato, pelargonium, dulcamara, and 1 liter of water if this is for infected area. Then we make priorities according to the ISPM number 6. We work on risk-based survey priorities. Let's say the first priority would be where potatoes at import and production, where warehouses and fields are. So priority number 2 would be for tomato in production, pelargonium at import and production, and appropriate time June, July. Then survey priority 3, if we still have resources and are available inspection units, then regions with surrounding dulcamara and irrigation water at the end of August will be sampled. This is an example of instructions for inspection on the field. As we selected the example of potato brown rot, we need to select other quarantine pests for annual surveys. In our project, a handy tool has been developed to assist in designing and planning of plant pest surveys. In the multi-annual survey planning tool, we combined several Excel tables which can support informed multi-annual and annual survey planning. It is used for designing and planning of plant pest surveys, where an enormous set of data have to be considered, including complex biology and epidemiology. The MASP table is based on scientific and technical knowledge of European Food Safety Authority, especially on their survey cards. The project tools for multi-annual survey planning can be efficiently used until EFSA applications for designing and optimization of surveys are developed. Those applications, RePest and OptiPest, are under the development, and even after they are completely developed, the output of our multi-annual survey planning tool can be used for import into the set EFSA applications. This was a brief introduction. To learn more, see our next videos on plant pest survey planning and reporting.